Hi, my name is Seema. The title of our PLRP project is Isolation and Mending of KRAS Mutations with CRISPR-Cas9 Nuclease. I will be going over the introduction and objectives. The KRAS gene mutation is predominantly a G12C point mutation responsible for several types of cancers. This single mutation leads to an overactive cell with heightened downstream effects causing abnormal cell proliferation. About 15 to 25% of non-small cell lung cancer adenocarcinoma patients test positive for this KRAS mutation. Although most NSCLC patients respond to EGFR and chemotherapy agents, improving their overall survival, patients with advanced stage lung cancer with KRAS mutation have a poorer prognosis. Unfortunately, no targeted KRAS treatment options exist for this small population of patients, so palliative treatment has been the ultimate goal of therapy. There is no defined treatment that exists for KRAS, but there is a genetic modifying technology known as the CRISPR-Cas9 system that has gained much recognition. Several studies have shown that this system can be used to engineer exogenous genetic information into an endogenous targeted genome. Donor DNA integration is accomplished through use of a chimeric RNA known as a signal RNA. Through the utilization of this system, the KRAS G12C point mutation can precisely be cleaved and replaced by the appropriate wild-type KRAS genome. The application of this genome tool into a KRAS G12C genome would allow further implications of the CRISPR-Cas9 to other disease states, as well as resolve the prognosis of KRAS positive individuals. And for the objectives, to expand the use of this tool for utilization in human diseases, we plan to use a CRISPR-Cas9 nuclease with this signal RNA to isolate the G12 KRAS mutation. The plasmid created will be inserted into a cancerous cell in hopes to knock out the mutation that triggers the spread of the disease. By successfully using CRISPR-Cas9 to detect and cleave the KRAS mutations, it would be possible to reduce the recurrence of other cancers which contain the same type of mutations by using the same method.
Hello, my name is Kaylin Caselli and I will be discussing the methods. There were two steps in this process. The first step in this process is understanding the molecular interactions between the signal RNA, which I will refer to as Guide 1, and the KRAS G12C mutant gene. A program known as Discovery Studios was utilized to visualize the crystallized molecular structures in order to compare the structural differences between the wild-type KRAS protein to the G12C point mutation. This program identified the key difference in the molecular phenotype of the proteins, which allowed for the visualization of genetic variants that can lead to mutations. The second step in this process is determining the efficacy of Guide 1 through creation of a cellular model. This process involves creating a plasmid using lentiviral Guide Puro and Guide 1 and inserting it into a vector. Once the vector is assembled, we will infect an HEK293 cell. Efficacy of infection will be seen through positive extracellular signal RNA secretion visualized via electrophoresis. This extracellular viral package will then be transfected into a KRAS cell with the G12C point mutation using type 2 CRISPR. Cleavage of the point mutation will be seen through electrophoresis again, thus demonstrating CRISPR-Cas9's ability to successfully reverse the phenotype.
Hello, my name is Tigris Heron, and I'll be going over the results section of this project. In the first step, we use computer imagery analysis to look at the crystallized structure of the wild-type KRAS receptor and compare it to the G12C mutant KRAS. In figures 1 and 2, you would see that there is a difference in structures. Figure 2 is missing an alpha helix, which is present in figure 1 of the wild-type KRAS. Now that we confirmed structural changes from our G12C mutant, we were able to find our signal RNA. The signal RNA that we used in this project is referred to as Guide 1. Guide 1 had a quality score of 85. This means that it can accurately bind to the G12C mutant found in DNA in vitro about 85% of the time. Figure 3 is a proposed structure of our Guide 1. There are 5 main features which are color-coded and enable the formation of a complex between the G12C KRAS gene and the CRISPR. The main colors to pay attention to is the red region, which binds to CRISPR-Cas9, and the yellow section, which is the G12C mutant gene that binds to Guide1. Figure 4 shows a specific interaction between Guide1 and the CRISPR-Cas9. And finally, Figure 5 is a complete mechanism of action in where the signal RNA binds to the CRISPR, forming a complex. The complex is able to identify the KRAS mutation found in DNA and cleave the mutation. This is what we would hope to happen in human genes.
Hi, my name is Baghdad Muslat, and I'll be going over the discussion and implications. Concluding with researchers at Massachusetts Institute of Technology, the CRISPR-associated Cas9 can be targeted to specific loci by signal RNAs. When this CRISPR-Cas9 complex is created, it allows for the cleavage of DNA at the target site. These models demonstrate the feasibility to delete the mutation out of the cell, ridding it of potential disease. Using the signal RNA guide design listed, the next stage in this process will be replication of the complex and transfection in a viral model. Some limitations to this study may be the sequences of the guides, the type of vector used, and the type of cell used. They may not always work well together. In future studies, different cell lines and guides may be utilized for optimal success of removal of G12C mutations. Implications of the study. Malignant cancer intensifies with the progression of abnormal cells amongst neighboring tissue. Different types of malignant cancers identify with KRAS mutations. There's a known correlation between the KRAS mutation and the G12C, G12D, and G12V genes on chromosome 12, where the KRAS loci is preserved in pancreatic cancer and lung cancer. Technological genomic modification to render the malignant cell free of cancer is still very unknown to this day. With the aid of a single RNA sequence, the CRISPR-Cas9 is a revolutionary tool proved to perform efficiently and effectively in detecting off-pattern DNA sequences within the bacteria and human genomes. The ability of this technology to detect and isolate specific gene sequences has led to the application of targeting cancer cells. Despite CRISPR-Cas9's potential of detecting mutations on off-target genomes and transforming current cancer therapy, there has been no advancement in utilizing this tool in human research. There's also limited research on the utilization of CRISPR-Cas9 on genomic modification of human disease such as cancer. This proposed implementation can redefine current therapy methods for specific mutations in the human genome. Using CRISPR-Cas9 technology will hopefully provide an innovative way to correct the mutation before cancer spreads. Perfecting this technology can allow doctors to implement genomic therapy in KRAS mutations in different cancers and diseases with mutations involving this gene. And this concludes our study on isolation and mending of KRAS mutations with CRISPR-Cas9 nuclease.